Welcome back everyone to MMBS111, Introductory Microbiology. This is mini lecture 7.3 and we are going to be focusing entirely on catabolism, or the breaking down of large molecules into smaller molecules. We have 7 learning outcomes for this mini lecture. First, list 3 basic catabolic pathways and the ATP yield for each. 2, summarize glycolysis. 3, describe the inputs and outputs of the Krebs cycle. 4, know where the Krebs cycle occurs in eukaryotes versus prokaryotes. 5, discuss the significance of the electron transport system and compare the process between bacteria and eukaryotes. 6, explain the differences between aerobic and anaerobic respiration. And finally, name 3 useful products of fermentation. So, catabolism. We talked about catabolism earlier in mini lecture 7.1. Catabolism is a form of metabolism that uses enzymes to break down organic molecules. We're talking about large organic molecules into their precursor or monomer molecules. Okay, cells are then going to use those products from catabolism to anabolize larger, more complex molecules. So remember, catabolism is breaking down large molecules into smaller molecules, and anabolism is building molecules from their smaller component parts. Catabolism is going to involve 2 electron carriers, NADH and FADH, and energy. So the energy that is stored in the bonds of those organic molecules will then be released as those bonds are broken, and we get their smaller precursor molecules. Both electron carriers and energy are needed in large quantities for anabolic metabolism, and those molecules are produced during catabolism. So catabolic reactions help to ensure that anabolic reactions take place. So how do we get these materials and energy that are necessary for anabolic reactions? Well, there are 3 main ways that cells use aerobic respiration, anaerobic respiration, and fermentation, and we're going to discuss each one in this mini lecture. You can see on the right that we have diagrams for each of these reactions and sort of how they take place. We're going to look at aerobic respiration first. Aerobic respiration are reactions that convert glucose to carbon dioxide and water, and in the process they produce energy, or ATP. Aerobic respiration utilizes glycolysis, the Krebs cycle, and the electron transport chain. So all 3 of these processes are used in aerobic respiration. It relies solely on free oxygen as the final electron acceptor. And this process is a characteristic of a large number of bacteria, fungi, protists, and animals. Anaerobic respiration is a form of respiration that is used strictly by anaerobic organisms and facultative anaerobic organisms. It still involves those 3 processes. So anaerobic respiration is going to involve glycolysis, the Krebs cycle, and the electron transport system. However, oxygen is not the final electron acceptor. Instead, anaerobic respiration is going to use other oxidized compounds as that final electron acceptor. So some will use nitrate, some will use carbonate, and some will use sulfates. So instead of oxygen, we're using a different electron acceptor at the end. Fermentation is the incomplete oxidation of glucose, and fermentation is used by many different groups of organisms Animal cells also use fermentation as a way to produce energy. Oxygen is not required in order for fermentation to happen. And organic compounds like alcohols and acids are the final electron acceptors. You'll also notice that there is a significant difference in the amount of energy that is yielded at the end of these processes. So aerobic respiration generally nets 36 to 38 ATP molecules. Anaerobic respiration can net anywhere from 2 to 36, depending on how far the molecules are taken through the respiration systems. Fermentation only yields 2 ATPs from every glucose molecule. So it's not as efficient. However, it does happen more quickly, and sometimes that is, and it happens without oxygen. So those are two key factors that can determine if an organism is going to ferment instead of go through aerobic respiration. Glycolysis is the first step in aerobic and anaerobic respiration. All three of these metabolic pathways are going to begin with glycolysis. Glucose is our beginning molecule, and it is a 6-carbon molecule. We're going to break down glucose into 2 smaller molecules called pyruvates. They are 3-carbon molecules. In that process of breaking down glucose into pyruvate, we are going to net 2 molecules of ATP as well as 2 NADH electron carriers. Then, if we continue on into aerobic and anaerobic respiration, that pyruvic acid or pyruvate is going to get modified and enter the Krebs cycle. This is also known as the citric acid cycle. The intermediate step is going to convert pyruvic acid into acetyl-CoA, which is a 2-carbon molecule. We're going to lose a molecule of carbon dioxide, and we're going to gain another NADH electron carrier. Acetyl-CoA will enter the Krebs cycle, and it's going to, that 2-carbon molecule is going to bond to the 4-carbon molecule that is found in the cycle and produce citric acid. Citric acid is going to be broken down a little bit. We're going to lose a carbon dioxide molecule, and we're going to produce another NADH from that reaction. So now we've moved from 6 carbons to 5 carbons. We're going to lose another carbon dioxide molecule and gain another electron carrier, NADH, and form 1 molecule of ATP. This is going to get us to our 4-carbon compound, and as we move from the 4-carbon to bonding with acetyl-CoA, we're going to get another 2 electron carriers, 1 NADH and 1 FADH2. So if you look there on the right, you can see these are our products from the Krebs cycle. These are total products. So we have 2 pyruvates that are going to enter this cycle from glycolysis. So this process is going to happen 2 times. And these products are the result of both of those molecules entering that system. So we end up with a total of 6 carbon dioxides being produced, 2 ATP molecules, 2 FADH2 molecules, and 8 NADH molecules. These electron carrier molecules are then going to be taken to the electron transport chain. And this is where the greatest amount of energy in aerobic and anaerobic respiration is produced. The electron transport chain involves special cytoplasmic membrane proteins that are going to allow electrons and protons to pass from each cytochrome protein within the membrane in sequence, and that movement of those electrons is going to generate some energy. In the final step, in aerobic respiration, oxygen will be the acceptor of that final electron. Energy is going to be released from those electron carriers, and it's going to be channeled through our final membrane protein, ATP synthase. ATP synthase is then going to take that energy and form ATP from ADP. So each NADH electron carrier molecule that enters the electron transport chain will produce 3 molecules of ATP. Each FADH2 electron carrier molecule that enters the electron transport chain is going to produce 2 ATP molecules. These hydrogen ions that are carried by NADH and FADH2 are also moved across the membrane, and they enter that space between the cytoplasmic membrane and the cell wall. And in that space, as we accumulate hydrogen ions, we create a gradient where there's more hydrogen ions and more acidity on the outside of the membrane than there is on the inside in the cytoplasm. As we learned previously in chapter 6, these molecules always move from high concentration to lower concentration. So these hydrogen ions are going to want to return to the cytoplasm. And as they move through ATP synthase and enter the cytoplasm, we get energy created by this proton motive force. And this energy allows ATP synthase then to phosphorylate ADP into ATP. In aerobic respiration, we're going to form oxygen and water. In anaerobic respiration, we're going to form things like hydrogen sulfide, nitrite, nitrate, sulfate, these non-oxygen molecules are going to form instead. So if you think about the number of NADH and FADH2 molecules that left the Krebs cycle, we had 8 NADH molecules. Each one of those can produce 3 ATPs. So that's a total of 24 ATP molecules. Each FADH2 molecule, and we ended up with 2 in the Krebs cycle, can produce 2 ATPs. So that's another 4. So we get a lot of energy produced through the electron transport chain. In anaerobic respiration, the terminal step is a little bit different, as I said. We're looking at oxygen-containing ions rather than free oxygen as the final electron acceptor. So this is an example of one way that might happen. This is nitrate reductase. We end up with nitrate, NADH, and then we're going to form nitrite, water, and NAD+. So the nitrate reductase catalyzes this reaction and the removal of oxygen from nitrates, leaving nitrite and water. Many bacteria are able to reduce nitrogen further, and these bacteria are really essential for nitrogen recycling in the biosphere. They assist plants with being able to use nitrogen from the atmosphere. All right, so after we form our 2 pyruvic acids, we can also go through fermentation. Remember, fermentation is the incomplete oxidation of glucose. So we're not going to go through the Krebs cycle, and we're not going to go through the electron transport chain. This process happens in the absence of oxygen. So it uses organic compounds as the terminal electron acceptors. Typically these are acids or other organic compounds. Very little ATP is produced as a result of this process, only about 2 molecules. This process is used by organisms that do not have an electron transport chain. Other organisms may repress the production of electron transport chain proteins when oxygen is lacking in their environment and revert to fermentation as their way to produce energy. This does not limit the growth of the organisms. So fermenting organisms typically increase the rate of glycolysis, and this allows them to grow at normal rates compared to other microorganisms. There are a number of products that result from fermentation in microorganisms, and many of these products we use. Alcoholic beverages are produced as a result of fermentation. There are also a number of solvents that we use that are produced. Acetone and butanol are a couple of those that we use that are products of fermentation. A number of organic acids like lactic acid and acetic acid, and then several vitamins, antibiotics, and hormones are also produced as a result of microbial fermentation. And that is the end of mini lecture 7.3. Make sure that you go through these questions here, that you can answer them correctly. Also go back and look at the learning objectives. Make sure that you can respond to each of the objectives, that you understand them, and that you're able to perform those things. For our critical thinking, state 2 ways that anaerobic respiration differs from aerobic respiration. In order to answer this question, you're going to need to understand both processes, aerobic respiration and anaerobic respiration, and find those differences when comparing those 2 forms of respiration. So that's the end of 7.3. I hope this video was helpful.